Hi everyone. So I'm Paloma and I will be talking about my PhD, but also about the project in which my PhD is included. So this is me. I did my undergrad in the Universidad Autónoma de Madrid in biology, and then I specialized in plant and fungal taxonomy, conservation and diversity at Kew. And finally, I went back to Madrid to do my PhD in the Royal Botanic Garden there. So we work here. That's our garden. It's really small comparing to yours. And my PhD is included in this project that is called the Odd Man Out project. And basically, we work with tropical plant diversity patterns in tropical Africa. So we mainly focus on the continental tropical Africa. And it's funded by the Spanish government. This is our team, as you can see, and also all the institutions that work with us. You can see Patricia Barbera up top. And a little bit about the concepts of our project. So we work with the odd man out pattern. It was described by Richards in 1973. And it was described as lower diversity in the tropical continental part of Africa than in other tropical areas. So if you compare with Madagascar, that is a special case, but also if you compare with tropical Africa or the neotropics. Tropical Africa, no, sorry, tropical Asia. And you can see there the graph. In the A, you can see what we called the tropical region in continental Africa. And you can see that the real tropical rainforest is a really small area. In the B, you can see a graph of the amount of species that you can see in the different tropical regions. And tropical Africa is also a small part of that. In C is more interesting than it shows. And you can see the proportion of genera and the amount of species that they have in all of the regions. And you can see that in Africa, you have more smaller genera and less big or medium genera. And finally, you can see in the D, the number of species versus the surface area of the tropical forest. And you can see that the pattern is there. So you can observe the pattern, but why is happening? We want to answer that. Classically, there are more hypotheses than those that you can see, but those are the ones that we focus in. The first is time to speciation. So it talks about that the lower diversity in Africa could be explained because the colonization of the species is more recent than in other tropical areas. And there's not enough time to accumulate that diversity. The second hypothesis is that this lower diversity could be explained for a more stable tectonical history. So from the paleogeology to now, you will see less orogenies and less movements of the tectonical plates, less accidents. That is not so correct nowadays because the latest paleogeological studies show that there has been a lot of activity in Africa also. Then you have the area for speciation one that implies that the lower diversity may be related to a reduction of the tropical zone from the paleoclimate till now. So you will have less area, so you will have less amount of terrain for diversity to occur. And then you have the evolutionary determinant one, that is a special one. So it's not focused on the climate or the area itself, but on the characters that the lineages in Africa accumulate. So they saw that some characters, like for example, dioici, accumulate more in the lineages of Africa than in other parts. And newer studies in Annonaceae and other families show that those characters that could be a handicap for diversity are not so important they are not causing so much extinctions or less diversity than in the lineages that doesn't show the characters. But we want to test it out with our model clades. And finally, you have the H5, that is the paleoclimatical instability. So this hypothesis associate the lower diversity that we can see with climate change events in the history of Africa. So you will have periods of aridification, or you will have dry, like really large dry epochs after cooling events, and so historically. And they can be linked to moments of lower diversity or even extinction in the past of Africa. So what is our model clade or our group of study? They are the Euphorbiaceae. It's one of the most diverse family, as you all know, and is not only diverse in terms of species or genera, it's also diverse in terms of habitats, morphological strategies, physiological advantages, and changes. And you can see that in that image. You have from trees to geophytes and cacti, and you have everything that you want or everything that you can imagine, you have it. And we selected 35 genera and 19 clades. Those clades come from the Burdak phylogeny of 2005. That is the one that you can see there, and they are marked in orange. So why we choose those clades? We chose them because they were diverse. So you have really big ones, really small ones, genera that are mega diverse, and others that are even monotypical. You have genera that are really diverse in terms of morphology or habitats, and ones that are really stable. And regarding morphology, we were focusing on sexual system, but also in habitat, and also in like strategies, different strategies that you could use. So which ones are the project goals? The first goal that we have is to increase the taxonomical and the taxonomic knowledge about Afrotropical Africa. The second is to infer the evolutionary relationships of these clades and the divergence times of all of them, and to see which patterns could emerge of each of the clades or genera that we work in. Then we want to examine the explanatory hypothesis of the odd man out pattern. So all the ones that I have showed you, we want to examine them with all of the model clades, with every of the genera. And finally, we want to synthesize all of this information to answer macroevolutive questions. But how are we going to do this? On the one hand, we are going to do a lot of field work because we want to complete a really big sampling. Patricia went to Gabon and we went together to Congo. But also, and mainly, we are getting all of our samples from herbaria and private collections. So we have been through all the world, all around, and one of the herbaria in which we work more is here in Missouri. But also, Patricia went to Tanzania, and we have been to all the main collections in Europe, like for example Brussels or Lisbon, yeah, Lisbon, Edinburgh. Also, we are doing morphological comparative studies. This was like a pilot study that started here at Missouri with the Madagascar specimens. And now we want to update that and include all the African species too. And we also want to do taxonomical reviews. But this takes time, as all of you know. And it's linked with the second goal. We also are doing UCN assessments to target the conservation part of the project. Then we get into the genomic part of the work, and we are using target sequencing or HiveSeq. That is a different kind of whole genome sequencing that you maybe know, and we are using the Euphorbia bait kit that includes 431 nuclear genes that are they are different from the ones that are included in the angiosperm one. They only match in like a really small percentage. And what is different about the HiveSeq? So on the one hand, you will start normally, like with the libraries and with the DNA extraction prior to the libraries, but then you will do an enrichment. That enrichment includes a lot of hybridization baits that are like mega primers. It's not the same technology, it's not the same chemistry, but what it does is that it will enrich the regions, your libraries in the regions that you want, that are your target. And when you sequence that and then you process the data, you will for sure have more sequences of that region that it's interesting for you and your group of study. And then when we have all the data, we will process it. And our goal is to make phylogenies and then to date those phylogenies to study the divergence of the groups. We do that with a really complex pipeline that includes a lot of convolutional processes and quality checks. Finally, you will have a species tree and also all of the gene trees that allows you to do a concatenated matrix, so like a super gene tree. And then when we have all the phylogenies and we have dated them, we will use microevolutive models to try to test all of the hypotheses that I have talked about. And those models are really complex, and depending on what data you are combining your phylogenomic data with, you will use one or another. Those are only an example, and for example, the one that you have here on the left is more focused on using characters with your phylogenomic data, and we will be using that to test for the dead end hypothesis with sexual system, for example. And what we aim to do is that we want to have our phylogenomical data and all of the other layers of data you can put on top. So like paleoclimatical data, sexual system data, characters, abiotical factors, all that you can think of. And we want to do complex analysis that allow us to test the hypothesis that we have set. And finally, we want to do a macro synthesis of everything. And this is the point in which our project differs from the classical approaches that you will find in biogeography. In biogeography, there's this fight in which you have either model clade studies with species that you really know, that are small data sets that you can answer microevolutive questions, and those other really big data sets or of different genera, even different families, of which you maybe are not an expert, but that allow you to ask macroevolutive questions. And we want to try to bridge them. So we want to do a lot of model clades that are related to one another, so within the Euphorbiaceae family. And when we have all the information about all of them, and we have studied their taxonomy, and we are working with experts in those clades, when you have all the information, you can analyze each of the clades. And when you have all of the microevolutive questions answered, as far as you can, then you combine all of them with different methods that I will talk a little bit now to try to answer your microevolutive questions. And which ones are those methods? On the one hand, you have Bayesian inference, and this is a BIB model that usually is used for Iceland biogeography. And you will have different datasets per one of the clades. For each clade, you will have a data set, and you will do a mega analysis of all of them, comparing their rates. So diversification rate, speciation rate, extinction rate, whatever you have, and all of the information that is necessary. So if you are putting all the datasets per island, you will use capacity of the island, distance, and so. We will be using, for example, number of species or the sampling percentage that we have. It depends on what is your data. And the other approach that is still a little bit, something really, really preliminary, is using machine learning models. So we want to train CNNs, so convolutional neural networks, to learn of the datasets that we have worked on, to predict larger datasets or datasets that you have not worked on previously. And that's a lot of things to do. Finally, some preliminary results that we have is our sampling. We are close to 70 to 80% of the targeted species in sampling. It has been an immense work. We also have more than 1,200 DNA samples, and we have sequenced at the moment 520 samples. So that's a lot of data, but we will be having more and more, because there's another PhD student in our project, and she is trying to complete our sampling. So as I said, we did some field work. We went to the Republic of the Congo, and we worked with the University Marine Nguabi personnel. And we went to the field. We returned with 247 numbers, and a duplicate of all of them will be coming here when we get our work done. And Patricia was the main PI involved regarding the Royal Botanic Garden and the Missouri Botanical Garden. So we are also working in conservation. We are collaborating with the Gabo Tree group of the IUCN assessments. So this group is also working with the garden, and we enter via Patricia, and we usually review each other assessments and do meetings in which we talk about our concerns, problems, criteria, and so. We have published these 4 assessments until now, and we are working in a really large data set that includes mainly edge genera, so monotypical or really small genera species. And these are some preliminary results. They are different than the one that you will see if you go to the IBC, because I'm working at that at the moment. But we have a preliminary phylogeny for the continental African species. It includes 160 samples, and regarding the sampling is more or less 70% of the species of Africa, continental Africa, and it has representation of Madagascar, Asia, and also the neotropics, because we thought that for croton it would be very important to see how the species come and go from the continent to Madagascar and back, because we know that it's not a linear history. And what we thought, or what we saw in the Sanger sequencing that we had, it's reflected also with the HypeSec target. So you can see that the continental African species doesn't form a monophyletic clade. They are quite dispersed, and Madagascar, the colonizations from Madagascar and back are constant. And it will be really interesting to do the biogeography studies of these species, but it's not done at the moment. It will be a future thing for me. And now I'm including more samples also. On the other hand, we have preliminary results about the the macro evolutive section of the project. So this is a preliminary phylogeny including 49 representative of all the lineages that we wanted to target. And you can see that we have represented the three subfamilies of the Forbiaceae. And what was really interesting to see is that most of the really small to medium genera, so 10 species or less, have their crownates before the cooling of the Paleogene. And you will see why that is really interesting. Regarding the pattern that I talked before, we say that it's that you can see a lower diversity in continental Africa, but we wanted to know how you achieve that, how you come from a tropical area to the pattern that we have now. And these are the preliminary results of the diversification analysis. And all the upper ones are done in COMET, so compound poison process in tests. That is implemented in R, and it's really quick to run if you want to give it a try. And the lower ones are done in REP-base. And they are quite similar, but the ones in REP-base include another dimension. They also have into account the percentage of sampling that you have for each of the clades that you are targeting. So they are including the bias that you have in your sampling. And also the computing process, the computing algorithm and how it computes, if you compare both of them, it's really different because both models do episodic birth-death models. That means that you have a time frame and you cut small windows in that time frame and you allow the rates to change in the windows. But the COMET one and the REP-base one treat those rates and those windows differently. So for the compound one, you will have exponential changes in the rates in all of the windows. And in the REP-base one, they will be linked. So they will be free to change, but they will correlate to the rates in the window before. So it's more cohesive. It's also longer to run. You can see in the first graph there that the net diversification rate, so the difference between speciation and extinction, it's stable, and then around 20 to 10 million years, it goes really, really low, and then it will emerge again. And that small window could mean mass extinction, but you will not know because maybe it's only low speciation. So you have the second graph that tries to measure only extinction. If you have worked in biogeography, you know that measuring extinction is really hard. But this is like an approach. It is an approach. You try to do that, and you can see in the same time frame, you can see that the extinction goes a little bit up. But then you will have the third graph that measures the probability of a mass extinction at that point, and in all of the windows. But you can see that the only one that is significant, so up 0.4, is the one that is between 15 and 5 or 15 and 10. That is the green line. The lower graphs that are from REP-base show a similar picture. So you can see in the first one the pool of the present, and the diversification, the speciation goes really up. But that's because that's an artifact cost that we know the diversity now, but we don't know the diversity that went extinct from later to now. So it shows that we have a lot of diversity now, but that's only an artifact. But you can see it's not so great with this color palette, but you can see that around the same time, the speciation is not going up so fast. And it's the same in the orange ones with the diversification, that it goes really, really deep. Then the blood, that is the confidence interval. And the line is like the middle value. And finally, you have the extinction, and in the same point, both of the analyses agree. At the same time, you have a really peak on extinction rates. What does that mean? If you put that into context with the paleoclimate history that we know, it shows that around 25 to 5 million years, you have a mass extinction happening or something happening. And related to climate, you can see that cooling went on in the Paleogene, and from the Miocene, you can see the cooling effect, and that cooling was accompanied by an aridification in Africa, and that aridification reduced the area of tropical forest, and that gives us like a hope of testing our hypothesis when we have a full data set, because without a full data set, this is only preliminary, and maybe we don't see the same when we finish. But now we are more closer to accept the hypothesis of the paleoclimatic consequences, and also the aridification one working in one both at once. It's not included in the presentation, but we are also doing niche ecological niche reconstructions of all the genera and all the clades per species also. And we are seeing also like movements within Africa that match the patterns of climate, paleoclimate that we know. And if you want to know more and to see further results, because they will be quite different, because now I have almost the full data set, so it will be a great update from that. I will be talking about that in detail in the IBC. And finally, I'm here doing a research stay in my PhD. I'm on my third year of fourth. And here I came to work with Patricia and also with Alex and with Irene. And I'm working mainly in the taxonomical review of the continental African species of croton. So I had a preliminary phylogeny, and now I have an extended one. And I'm reviewing all of the materials that went into the phylogeny and that were selected for the phylogeny, even if we don't have sequences. And we are comparing them with other materials that we have, and also working with all the floras, all the descriptions, all the information that we know, to try to re-identify them, to update our phylogeny, and to, in a future, do a revision of all the continental African species. And that will take a lot of time. And Patricia Barbera and Ricarda Rina are involved in that. And I don't know when it will be published, but it's a really nice work to do, and I'm loving it. And also I'm here making digitalizations of pictures, more like of all the specimens that we are interested in and that we have samples of, because in the future we want to include the characters, the morphological aspect of the species in our studies, not only for the taxonomy, but also to compare with the phylogenomical data with the models that I have shown you. And that will be an update on the study that they started in the Madagascar species of croton, and we want to include all the African ones. And that's everything. I don't know if it was too quick or not, because I didn't put an alarm. I'm sorry if my English was a little bit rusted. And this is like a pre-rehearsal, pre-IBC. So if you have any questions, I will try to answer them.